Greetings, germophiles, and welcome to another edition of microTalk. This is Carl Close. I'm from the University of Texas in San Antonio, and I want to introduce first the microTalk team. So if you raise your hand right there, that's Dr. Jesus Romo.
Hi, everyone.
You want to introduce yourself?
Yeah, hi. I'm an assistant professor in the Department of Molecular Microbiology and Immunology at UTSA, and my work focuses on polymicrobial interactions, specifically between bacteria and fungi and the human gastrointestinal tract.
Okay, and so to his left, we have Dr. John Mekalanos, but apparently he's brought somebody with him. Who do you have with you?
Hey, germophiles, this is microTalk, the awesome undergraduate student majoring in microbiology and immunology at UTSA. What's up, y'all?
So apparently the puppet is in the house. So this is a very exciting occasion for everybody because usually you don't get to see microTalk. But we are very excited today because we have royalty sitting on the stage right here. So we have the current president of the American Society for Microbiology, Dr. Virginia Miller. Well, starting at the end of this meeting, right? Oh, July 1st? I apologize. She's a wannabe president. But almost, we'll call you quasi-president, okay? So Dr. Virginia Miller from the University of North Carolina at Chapel Hill. You want to say hi, Virginia?
Hello, how is everybody today?
So we're very ecstatic to have her. And we also have the president-elect, and that is Dr. Theresa Koehler, and she's from University of Texas in Houston. And you want to say hi to everybody?
Howdy, everybody. Welcome to Texas. I'm from right down the street, and I'm really happy to be here today.
So we have these fantastic guests here. I'm sure you all are interested in their perspectives on ASM and things like that. But we want to talk a little bit about the science that they do and what actually drove them to be the presidents of the Society for Microbiology, because they're actually microbiologists themselves. So starting with Virginia, I think we'll talk a little bit about plague. So Dr. Miller is a well-known and respected scientist in the field of plague biology. She studies other gram-negative bacteria as well, but let's talk a little bit about plague. So what made you decide to study Yersinia pestis?
Well, to be perfectly honest, it was a postdoc that said, we have to do this. So I always listen to my postdocs because they're all smarter than me. And so we started working on plague, but honestly, I think it's one of the most interesting bugs you could ever work on.
Absolutely.
Yeah, so, I mean, this is something that I've been very interested about. I'm not a plague biologist, but I think it's playing a very significant role in human history, right? Can you talk a little bit about, a lot of people believe and think that the rat was one of the main carriers for the plague, but I think there's other ideas out there, right, about other potential vectors. What do you think about that?
Well, to be perfectly honest, I think the rat was a main driver, and I'm not really prepared to talk about other alternatives at the moment.
Yeah.
The rat is king, right?
Yes, but we'd rather it wasn't.
So, Dr. Miller, you also work on other interesting pathogens like Klebsiella pneumoniae. So what made you also, start looking into Klebsiella compared to this wonderful organism that causes, plague?
Okay, well, I started working on Klebsiella because I wanted to try something different, and I wanted to start working on something that was understudied. And so I chose Klebsiella. And you might ask, well, why Klebsiella besides being understudied? I am a molecular geneticist in my heart of hearts, and so I had certain requirements on what I would want to work on. One is that I could do molecular genetics, and since Klebsiella is related to E. coli and Salmonella, I thought it would be easy. It was not, but it was possible. And then I also, being someone interested in pathogens and how they cause disease, I wanted a bug that I could, A, grow quickly, and Klebsiella grows really quickly, and I needed an animal model to validate that what we're studying is of interest in terms of pathogenesis. And so Klebsiella kind of checked all those boxes, and it's been really fun.
Well, and it's also, I would say, more relevant to current Americans than plague, right? I mean, plague killed lots of people in Europe in the 1200s, but
Yeah.
Klebsiella is killing a lot of people right now.
Absolutely, and that is another main reason to do it. And like I said, it's understudied. There's a lot to do, and so anybody out there is interested in picking up a new bug, I would recommend thinking about Klebsiella.
Right, and so Virginia, can you tell the audience a little bit about the growing problem with carbapenem resistance? Because that's basically the driver of the concern with Klebsiella, right?
Yeah. So Klebsiella is one of the most prominent carbapenem-resistant bugs that we isolate in the hospitals, and it's causing a huge amount of problems with that, in part because not only is it resistant to carbapenem, but it's resistant to a lot of antibiotics, including other extended-spectrum beta-lactamases, and now we're seeing ones that are not only resistant to carbapenem, but also colistin, and so it's becoming very, very difficult to treat, and its mortality rate and disease morbidity is very high, especially in hospital-acquired settings.
Yeah. So we're super concerned about that particular organism. Another organism that we're very concerned about is Bacillus anthracis, because of previous attempts at bioterrorism, essentially using this organism. And we have sitting here to my left a world expert on anthrax, Dr. Theresa Koehler. You want to tell people a little bit about what are we concerned about right now with Bacillus anthracis?
I think I'll step back a little bit and say that when I began working on Bacillus anthracis, it wasn't the concern, of course, that it is now. And I think that we don't maybe have very specific concerns about anthracis now that are really, really frightening and critical, although it's always a concern. But I think that studies of that organism and what we went through in 2001 opened our eyes to the potential for all kinds of what we now call select agents and the possibility that they could be used and cause great harm.
Right, exactly.
Could you talk a little bit about what makes this organism so scary, right? It's the virulence factors or maybe pathogenicity mechanisms that it uses?
Yeah, I think the biggest reason that there's justified concern about anthracis is because it's a spore former, right? So it can lie dormant in the environment, in the soil, and other environmental niches for decades, right? And if someone were to use this for nefarious purposes, you could produce Bacillus anthracis spores and keep them in your pocket for 25 years, and so I think that the ability to produce this organism easily, it's easy to grow, and to store it for long term in high numbers is probably the biggest concern, but it's on the select agent list. I mean, there are plenty of organisms, unfortunately, that we consider biothreats, but I think that the ability to make these spores and the fact that they're so long lasting and viable is probably the biggest reason that we're fearful of Bacillus anthracis. However, we do have a functioning vaccine. Right? So it's been used in animals for a very long time, for decades. In the former Soviet Union, people were vaccinated against Bacillus anthracis. We use the vaccine for laboratorians and for people who are otherwise at risk, whether they're veterinarians or farmers in an area where there is concern. The issue with the vaccine right now is that the immunity is not long-lived and you need frequent boosters.
But there's a recombinant form of the vaccine that's in preparation, right?
Right, yeah. There's been a lot of work. I mean, one great thing that came out of the unfortunate events of 2001 was this heightened increase and funding, to be honest, for work on Bacillus anthracis. And prior to that time, I'm kind of an oddball in that I've worked with Bacillus anthracis since I was a graduate student. So my whole career centered on primarily this one organism. Initially, when I started my own lab as a faculty, I was just one of a handful of people, and I was interested because I love bacterial physiology and genetics, and I thought it was a cool organism. But when that event happened, people with all kinds of expertise and ability and interest followed the money, which is completely appropriate, got into the field, developed relationships, and a lot of progress was made, including ideas for new vaccines.
So apart from the availability of a vaccine and hopefully some new ones, down the pipe, if someone were to get anthrax right now, what are the treatment options available?
So fortunately, unlike other organisms, Bacillus anthracis is sensitive to a wide variety of antibiotics. So this is not a multi-drug resistant organism. Ciprofloxacin was used in 2001, and there's an interesting story I've been told about Cipro. When the first Gulf War began, many, many years ago, there was concern about biowarfare. And so people thought about, well, if this were to happen, what would we use as antibiotics, particularly against Bacillus anthracis? At the time, ciprofloxacin was a relatively new antibiotic, right? So many things. At the time, penicillin was used, tetracycline, all kinds of common antibiotics could be used, but Cipro was very new. And the thought was, well, if whoever intentionally released this organism had manipulated it so it was resistant to common antibiotics, Cipro's new. They may not have done that. So that's the whole history of why Cipro was the big antibiotic in 2001. But to be honest, many different antibiotics could have worked just fine.
Dr. Koehler, I am 21 years old and I freak out about a lot of things. One of the things that I'm kind of like freaking out right now is that there's a possibility that environmental strains or environmental location of Bacillus anthracis can pass as toxic plasmid or the capsular plasmid to other rhizobial species. And I also love working in my garden. Should I be worried about it?
That's a great question. And primarily because I love thinking about environmental exchange of DNA, right? And so first, let me tell you, you should not worry about it. There's all kinds of genetic exchange happening against, for all microbes in the environment that we're not aware of, but we're becoming aware of as we isolate and sequence DNA from more environmental organisms. Bacillus anthracis is closely related to Bacillus thuringiensis, as you may have heard, and also Bacillus cereus, which are common soil organisms. Most strains of Bacillus cereus are not pathogens. A few cause mild food poisoning. Bacillus thuringiensis, which is another close relative to Bacillus anthracis, is actually a pathogen for some insects, for Lepidoptera and other insects. And so we actually use, if you're a gardener, you may go to your local gardening store and pick up a bag of BTI, which is Bacillus thuringiensis israelensis, and it's a natural insecticide. So Bacillus thuringiensis israelensis, you can sprinkle that on your garden and get rid of some critters that you don't want. So I wouldn't be concerned. Bacillus anthracis is in the soil. It's on every continent, but it's pretty difficult for a gardener to acquire it.
Oh, that's fantastic.
Especially a puppet gardener, I would
Yeah,
imagine.
exactly.
Yeah.
That puppet's doing a lot of gardening, apparently.
No, I make sure that I wash my hands properly. So
in thinking about vaccines, Virginia, so I understand also that there is in development a vaccine against the plague as well, right?
There's been work on a plague vaccine for a very long time, and
Yeah.
I think that the difficulty that people run into is that they'll get a candidate that's good at protecting against bubonic plague, but not against pneumonic plague, which is what you are most worried about in terms of a potential biothreat.
Is that right? So is it because of the route of administration? In other words, if it comes in through the lungs, it's difficult to protect against plague? Hard to say.
Yeah, I don't, I'm not an immunologist. I'm not really in the vaccine field, but, it has been true for many candidates. I don't know if there's some candidates out now.
Okay.
I haven't worked on plague for a little while, but there may be newer candidates that are working for pneumonic plague or are thought to be.
Yeah, well, I'm just trying to put the audience's, minds to rest that we have vaccines in development against these two dangerous organisms.
The advantage about Yersinia pestis, kind of like anthrax, is that it is susceptible to antibiotics. The key there, though, is that it is such a rapidly progressing disease that you have to know you have Yersinia pestis soon enough for the antibiotics to be administered and be effective. Because if you wait too long, it just doesn't matter. And that's the problem. And the early stages of the disease are like having a common cold. And so, for someone to go in and say, I might have Yersinia pestis, give me some antibiotics, it is probably not going to happen in time necessarily.
You mentioned two types, right? Bubonic and mnemonic. What are some of the main differences? Is that the acquisition route or?
Yes, definitely. So bubonic plague is the kind of plague that you think about initially from the Middle Ages, where people would have a big swollen lymph node under their arm or in their groin, and it was called a bubo, and that's where you get bubonic plague. And that's when you get transmission via a skin bite from a flea that's an infected flea, and so you get transmission of the bug, homes into the lymph node and grows there initially, and eventually spreads systemically, getting into the lung.
Yeah.
And then you have a lung infection, and that can be then transmitted directly person to person. You don't need a flea vector. And that's when you get primary inhalation of pestis, and it grows very rapidly. It loves the lung environment.
Right.
So what made Yersinia to love the fleas to be transmitted by fleas? Why did it not pick maybe a cockroach or a mosquito? What is the specificity for Yersinia and the fleas?
I think it was just a match made in heaven.
So I think that the audience would probably like to get to know our chiefs better. Let's just put it that way. So Dr. Miller, can you tell me a little bit about your trajectory towards becoming, who you are today? In other words, like becoming a scientist that studies plague. What were some of the critical events along your pathway that led you into this profession?
Oh my gosh, I think I've just been really lucky all along the way. I was always interested in science, and in college, I had no idea what I was going to do after college, just to be really upfront. But I loved biology and I loved genetics, and it was just the dawning age of molecular biology and molecular genetics, and I got hooked on it completely. I just loved the puzzles that you could use to figure things out, and I didn't want to go to medical school, and so I applied to graduate school. I had a TA who encouraged me to do that, and I got in, and I just took that leap, and it just felt like home when I got into graduate school, and it just never looked back.
Yeah, so Virginia and I share a lineage
Yes.
together, and so she was the first. It's true, right? You were the first PhD student of John Mekalanos.
Yes.
Yeah.
Yes.
So very when you're in the Mekalanos lab, you have to look up to Virginia because she was the first.
It's hard to look up to me though because I'm so small.
Well, when you're sitting.
So short.
Anyways, she shares a lineage, and there's a lot of history associated with Virginia being in John's lab, and I can imagine that was very inspirational to have the intellect of John Mekalanos focused on his PhD student, right?
Oh, it was so fun. We shared a bench for some time, and I'm a pretty neat person, and
John's not.
is not, and so eventually I had to put a piece of tape down the middle of the bench because he would spill over, and I said, if you spill over, I'm just pushing it away. So, John is an amazing mentor, and it was great to be able to be there with him at that early time, and all the wonderful people that were there as well, as like Ron Taylor and Marcia Betley, and people that came immediately after me, like Victor Rita and Carl here and Jim Slough. Too many to name, really. But yeah, it was a great environment, and I got really lucky to be there at the kind of ground floor of using molecular genetic tools to study pathogens, and I think that has just revolutionized studying bacterial pathogens.
Yeah, and hopefully, the younger people in the audience get some inspiration from that, right?
Yeah.
So you can be the next Virginia Miller.
Yeah.
Dr. Koehler, what was your first failed experiment, and when did that failed experiment become okay or got successful?
There are so
Because
many.
I've had way too many
She
failed
didn't want to be put on the
experiments.
spot, and here
There are
little
so many
micro
failed
session.
experiments to choose from. a few minutes ago, you asked me about genetic exchange in the soil. And so when I was a graduate student, I studied natural means of genetic exchange between closely related bacteria. And so actually, I do know my first really, it could have been failed experiment. So I was looking for conjugation, mating between different species. And I had a particular culture going. It was a co-culture, same flask, shaking. At the time, we had these big New Brunswick floor shakers, right, in environmental rooms that the entire department shared. And I had my flask on there one evening, and I came in the next morning to examine what had happened overnight, and someone had turned the shaker off. And I was really not happy. And so as a student, I could have just autoclaved the thing and repeated it, but I went ahead and we were geneticists, so we were doing plating techniques, following markers to look for genetic exchange. So I thought, what the heck? I plated it. And actually, after many previous attempts to get these two particular organisms to exchange DNA, I saw it for the first time, and the key was it was a static culture. So could have been failed, and I never found out who turned off the shaker, but I really am indebted to that person. It's been many, many decades now, but that was a good thing.
So technically, there's no failure in science.
I love that. Yeah.
Okay.
I mean, negative data is always very
That's true.
important, right?
I mean, that's what I keep telling my boss, but he doesn't want to listen to me. That every failed experiment is a success.
Yeah. I love that story, and I think we all have a someone turned off the shaker story. And I'm willing to bet it's the same person just going around random labs, just turning off people's things. But I wanted to ask you both, and you can address it in whatever order you want. Looking back in your career now, when you were a trainee, like a lot of the people here at the conference, what is maybe the one or two things that you wish someone would have told you as you were embarking in this journey that has been helpful or a big realization for you?
I'll answer that. Somebody should have told us all how much fun it is and all the great people you're going to meet along the way. That is really what it's all about, I think.
Spoken like a true president of ASM.
Yeah, I completely agree. Yeah.
I totally agree with Virginia. It's all about the people, the colleagues that you meet, and every day is different, right? I use the term discovery science. I can't stand the term basic science. My young adult children tell me is a horrible word, and I agree. It's every day you're discovering something. It may be negative data, but it's a new thing, right? It's to me, it's the most exciting career that I could imagine.
Right, and empowering too. I think that's a good word for it. When you learn something new every day, you kind of feel empowered to continue, right? So since you both illustrious scientists are going to be leading the ASM for the next 2 years, what are some of the ideas that you have? Or do you have any about advancing or changing anything about ASM? Or it's a steady ship, and you want to just keep going the way that it is?
Well, I talked a little bit about it the other day.
Yeah.
We have had this big envisioning plan of what the future is going to be like for ASM and society and its role in society. And that's something we're really working hard on. I mean, it's a big question. We want microbiology to be part of the future and making sure that we all have a better future. And I think microbiology can have a big role to play in that. And so we want to think about how ASM can help microbiologists and help society do that and play a role. And so that's a big question that I can't do by myself. I need Terry, I need all of you, and we're going to make it happen because I know we can.
Yeah, I my first ASM conference was in 2017, and I've seen so much growth, and I've seen all these initiatives having to do with inclusiveness and diversity. Where do you two see ASM going in that area of diversity, equity, and inclusion?
I think microbiology and the ASM is really already a leader in that. I mean, if you look around, we are one of the most diverse professional organizations that exists in science, or I would say in other fields too. So I'm very proud of the ASM for what they've done. That doesn't mean we are finished, right? We have a lot of work to do, but there's a lot of thought is continually being given to these efforts. I'm sure that we will just continue to advance in that area. just to answer your question a little bit in terms of what I would like to see and what I think everyone in leadership and all of you would like to see is that we just went through an exercise with the pandemic where our eyes were open to how important public trust is in science. Mm-hmm. And I think that we need to work to enhance that trust, and not just trust, but confidence in our ability as microbiologists to advance approaches that are going to make life better for everybody. And I think, and one really cool thing that I've noticed at this meeting is the attention to microbiology and climate change, global health, and personal interactions. And so I think we're really on the right track for that, and we have to keep a keen interest in advancing. We want to be known as the go-to place for microbiology or anything remotely related to microbiology. If anybody, any scientist or non-scientist has a question, they should go to the ASM and be able to get that answer. And so I think that's to me that's exciting. I'm looking forward to working towards that direction.
Yeah. One of the most, in anyone in the audience who doesn't know this, one of the most amazing things about Dr. Koehler is that she was essentially retired and came out of retirement to lead this organization. And that's the passion and dedication that she has towards the ASM. And so, I think it's really inspirational to me, and I think to everybody, that there's this passion among the scientists that are leading the ASM to make it, like you said, integral to, our lives or whatever. So
yeah, and I'm sure for everything that you mentioned, the trainees and the younger scientists play a very significant role as part of ASM. Do you have some words of advice for them as getting involved with the organization and continuing to support its growth? It could be.
Well, I recommend everybody get involved with ASM. There's a place somewhere in ASM for you to get started and to do something that you're passionate about, and you'll meet new people and interesting people and accomplish big things if you get started doing that. There's a volunteer button on the website. Go check out what's available and give it a try. I know I got involved at ASM very, very early in my career, and it's just been a great experience all along. Obviously, since I'm still here, I didn't give up. And I think ASM has just been a wonderful community that you can grow as a leader, as well as a scientist and as a person, and take advantage of it. It's there waiting for you.
So if both of y'all are or were or are TikTok influencers for microbiology, what will be the area of microbiology that you would be influencing right now? Like even if it doesn't,
you've heard of TikTok, right?
Yeah.
I have, but I am not on TikTok.
Just what kind of, what area of microbiology that you would be influencing? Somebody like me, I want to do microbiology for the next 40 years. What will be the one video that I will see and inspire myself to go after? It doesn't have to be your pathogen or your area of research in microbiology. It could be something else.
Could be a dance video, maybe.
Yeah,
maybe.
I think for microsexual, that's easy, the TikTok, because he studies Borrelia. So ticks, there you go. But for the rest of us, it's a little bit more challenging. I mean, I like that question, though.
I don't know.
See? That's the kind of question.
The puppet,
it would have been helpful
stumped
if
the
we
president.
had the questions ahead of time, and we could have thought of something really brilliant to say. But
Could have done a dance.
we could have.
That's a dig at me. Because they asked repeatedly for questions, and I didn't give them any. So, anyways.
So none of this is our fault. The first sign of leadership. It's not your fault.
I did want to ask Terry. So this is your hometown, and there's a lot of people here who have never been to Houston, and they don't know anything about this place. Can you tell them what makes Houston special for science, for studying microbiology, for example?
So you may have not made a trip down there. It's a 15, 20-minute train ride. But we have, in Texas, first of all, I will say that largest is a synonym for best. And we have
that's absolutely true.
And we have the largest medical center in the world down the street. It's what brought me here. I was an East Coaster, born and raised, loved the East Coast, never thought I'd leave. But here's a side piece of advice. If you're looking for a position, try not to be too geographically limited. And I tried to have an open mind. I interviewed here thinking I'm never going to move to Texas. And here I am after 32 years. Anyway, the medical center here is world-renowned for research and patient treatment. And it's just, and there's a ton of microbiology going on there at a variety of institutions. It's just been a fabulous place to work. Houston itself is also a great city. We are an very international city. We're one of the world's largest ports. We have a very diverse community. It's just, I'm just going to say that as an East Coaster, I surprised myself and fell in love with Houston. I think it's a great city.
And there are a lot of nice places to go and eat.
We have really good eating here.
Yes.
Yeah, they're actually well known within Texas as having the best restaurants actually in Houston, and that's no lie.
That's the truth.
And I have to say that, I'm from San Antonio, and I really don't spend any time downtown Houston ever. And it's actually really quite nice. I was kind of surprised. Coming from San Antonio, of course, right?
I'll take that as a compliment.
It's a compliment. It's a compliment.
Yeah, it's too bad. This is a little bit unusually warm for us this time of year.
Yeah.
But if you can stand 10 minutes outside, Discovery Green, which is adjacent to the convention center, is a beautiful park. It's a really nice place to walk around if you have, can take off your jackets and enjoy a little bit of time outside.
And put on your shorts, right? Hey, Virginia, so can you tell us a little bit about what makes North Carolina and specifically Chapel Hill like a great place to do science?
There are a lot of things that make it a great place to do science. The weather's a little cooler, for one thing. But also, it's got a temperate climate. We've got wonderful people that are really interested in working with each other and thinking about not only the research they're doing, but really getting involved in the education and training and thinking about the next generation. And then, of course, we have that other school up the street, which will not be named, that also has some wonderful microbiology, as well as NC State. And also Wake Forest is not far away either. So we really have a lot of really great universities that have a lot of great science going on. And then, of course, there's Research Triangle Park that is doing a lot of really wonderful work and a lot of different opportunities. And a lot of people come and they love it in North Carolina, and they stay and they find interesting jobs doing microbiology. So,
well, I like the pitch for both places. So I think, going back to the trainee experience, right? When you were deciding where you wanted to do your PhD, I'm sure some of them are thinking about that or where they're going to go do their postdoc. What were some of the factors that played a role in your decision, or what are some tips that you might have for them as they're coming up to make, which is a big decision in their life?
In addition to keeping an open mind and taking advantage of every person, every situation that you're in, and getting to know that situation, that person, making connections, I would say you need to be a little bit brave and put yourself out there. So I'll just tell you my little funny anecdote is that I got my postdoc, which was an amazing postdoc that I still feel like I didn't deserve, because I gave a talk at ASM, at what used to be called a general meeting. So I was giving a talk at the general meeting of ASM a million years ago, and somebody walked up to me after my talk who was a huge leader in my field, so John Collier. I'd never met him, but I had read all of his papers, and he offered me a position after my talk. And I had applied to other labs, but right away I said, oh, okay, that sounds good to me. So take every opportunity you have to show your work, whether it's a poster or a talk. Get yourself out there and meet and talk to everybody you possibly can.
That is fantastic advice.
So when both of you all were undergraduate students, did you all have any amount of anxiety about your potential future and your success? And how did you overcome that anxiety? I mean, as an undergraduate, you must, or you guys did not have any anxiety at all?
I had no idea what I was going to do when I graduated till very late in the game. And I think I mentioned earlier that I had a TA that really encouraged me to apply to graduate school, which had never been on my radar. I had never had a faculty member or somebody say, you look like you have the ability to do this. And it was just not in my brain. And my parents didn't go to college, so it just wasn't something there. But I took the leap and did it, and I have never looked back because it was absolutely the best advice I ever got for me.
It's awesome. So we have the opportunity, if anybody has any questions for either Dr. Koehler or for Dr. Miller, we have a microphone. Does anybody, if you ask a question, this is what I always tell my class too, you'll get a prize, right? So we have these squishy little microscopes that are hot collector's items. And if you ask a question, you will get a squishy microscope. So anybody want to ask a question of either illustrious guest that we have here?
Hi there. Can you address what you think the impact of artificial intelligence and machine learning is on your particular fields?
Oh, gosh. You didn't want to start easy with like a softball, a microscope or something. From what I understand, not being, hot on that field, is that it's really going to help with data, help mine through data analysis. And I think definitely if anybody's heard the talks that came out of the retreats for HMB and MBP or EEB groups, that is really what they think is important. And that's sort of how we want ASM to be able to help all of us, is to help get some valuable training on how to use AI to help us make our science progress better and to use the most of all the data that we had. So I'm hopeful that it'll be a good thing, but it's also something we got to be a little careful about too.
Who else has a question? Oh, go ahead, Terry.
I 100% agree
Terry.
with everything Virginia said. I will just add that I'm thinking maybe a little bit more outside of the box. And imagine if, so I don't know, how many scientific papers can you read a day and really get a deep understanding and be able to have the time to think deeply about the experiments? 2? Yeah, 3 tops, right? And I'm brain dead. What if you could use AI to read 1,000 papers in a day and generate hypotheses for you based on what was in those papers? It's pretty awesome to think about things like that, right? We read papers, or at least in the research field, to generate to figure out what's missing there, what are the gaps in knowledge, and what are some hypotheses. What if you could have AI do that for you, and then you figure out the best way to address the hypothesis? Kind of cool to think about.
That's a positive spin, right? Because as an instructor, when you're teaching class, for example, all the instructors are fearful of AI because it makes the job much harder, right? But I like that. So it's a very optimistic take on AI. Sure. I like that. Virginia, did you have something else to add to that?
No, I'm going to skip that one.
Okay. Who else has a question?
Hi everyone. Good afternoon. I'm Ana Villanueva. I'm from the Philippines. This is actually my first experience here at ASM Microbe.
Nice.
Thank you for the wonderful talk. And I just wanted to ask, in your very successful careers, have you ever engaged in global health collaborative research, and how has it, helped your career? Because for me, I'm here because I'm looking for potential mentors and collaborators. But what is in it for people like you guys who have expertise in these fields?
Nice question.
Well, I would call my recent experience in that way sort of a peripheral global health question, because it's with our work with Klebsiella. And it's a big problem all over the whole world, and there's a lot of variation in Klebsiella strains and their ability to cause disease. And so being able to get a good collection of strains from all over that are diverse is really helpful. And so I was able to collaborate with a group from Australia that was involved in that, and it's been really, really a nice thing to do. It's not that easy to get well-characterized strains with clinical histories attached and genomic sequences available, and so that's a really, really nice way to have a collaboration or get one started.
Well, welcome to Houston. That's a long trip from the Philippines. I did the exact reverse just a few months ago. This is a long trip. So nice that you're here. Yeah.
Carl, I'll put in a plug. If someone opens up a podcast app and subscribes to microTalk and shows me, you can have one of those stuffy microbes. So MicroTalk is a podcast we produce at ASM. What do you cover most, Carl, on MicroTalk? Why would they listen?
You should all listen to microTalk. Absolutely. We cover microbiology, and we talk to scientists that actually work on these bugs and study microbiology. And we talk to them as scientists, but we also talk to them as people. And the goal is to personalize scientists so there's no mystery around it. But the one thing about scientists that is that they're most passionate about what they work on. So we found it to be very inspiring to chat with scientists about what their passions are. And so I encourage you all to listen to MicroTalk. And there's some really great hosts of the podcast that that are up here on the stage.
Yeah, if you're an undergraduate, you'll get a lot of wonderful ideas as to what you can do with your life.
Yeah, and I think we really have tried hard to have a good collection of microbiologists from different walks of life, different backgrounds. I'm a mycologist, so I will say that we have a good collection of mycologists that we've interviewed, and that's something that you don't really see. So I would encourage you to give us a listen, give us a chance. I think it'll be something that you'll enjoy.
Yeah.
Hi, I'm from India. This is my first time attending this conference, ASM Microbe.
Welcome.
And this is my international visit, first time in my lifetime.
Wow.
So I'm happy to be the part of this one. As a woman, after joining this ASM conference, I came to know that ASM Connect, it is very useful for connecting with people. So daily they are sending the message, what type of work, what type of people to be connected. So I'm willing to be a part of that subcommittee member. So what is the procedure? Do you have any idea since you are a president of that one? So this will be the right time to ask, what is the procedure for being a subcommittee member from my place?
Basically, if you go online to the website, for the ASM website, it's maybe not, it may be buried a little bit, but there is a place where are you interested in volunteering?
Yes.
Okay? And you'll see that on the website somewhere. You click on that, and it gives you a list of all the different groups and subgroups that are always looking for members. And it also gives you a form to fill out about your interests, to try and get a good fit for you. And so we collect names, and when we need people, we reach out to people who we think might be interested and see if they want to get involved. Does that help you? Okay. So do it.
Do it. Okay, last question. Ray's wanting us to wrap up here.
Hi. So I teach undergraduates how to communicate in science, and I'm wondering, as infectious disease biologists at every stage of our career, how do we build trust with the public? So even from undergraduates to graduate students, how do we work on that?
Listen.
Listen, that's number one. I totally agree. And we should always need to be transparent, and just very communicative. And I think it's also important to include in any message you want to give in terms of information to somehow work in the scientific process that led to that discovery. I think that's very important.
Which one's measles? No. I'm not a virologist. What can I say? There we go.
It looks exactly like that.
Okay. Well, it was a pleasure, doing this podcast with Dr. Miller and Dr. Koehler. Both are extremely inspirational scientists, but also friends. And I really enjoy that aspect of the podcast is we get to talk to people that we really like, and we get to expose exactly what wonderful people they are. So I really want to thank both of you for being here today. Little micro session. Do you have any last words of advice?
Oh, there's one question that I failed to ask. Is what would you have been if you were not microbiologist?
You can't ask any more questions. Ray is going to
okay, well, sorry.
chop your puppet off.
Yeah.
Okay, well, then I'll end it. And,
Jesus, you got any?
Yeah, I really enjoyed this, and I think I am very happy that we have you in charge, and it makes me very optimistic about the future of the organization. So thank you so much for your hard work.
Exactly. ASM is in good hands, at least for the next 2 years. So thank you all for coming. We appreciate it.
Thank you.
Thanks to our hosts.
Thank you.
Thank you very much.